Good evening, everyone, and welcome to today's session. We're gonna start in 5 minutes so that people can join in. Okay.
इंडिया में पहली बार एक आईआईटी ने
Okay. So we can start our today's discussion. So if you remember what we were discussing in the last class, we started with the protein's secondary structure, and particularly we were discussing about alpha helices, which is a type of predominant secondary structure present in biological system, right? So as there is a possibility of both right-handed and left-handed alpha helices, but in analysis it is found that there are more prevalent is the right-handed alpha helix. So this is a old paper, like in 2005 somewhat. So they actually analyzed all the alpha helices present in the structural database, and then they made this analysis that what sort of alpha helices are predominant. So we'll go through this abstract very quickly. Okay. So all naturally occurring amino acids, with the exception of glycine, contain one or more chiral carbon atoms and can therefore occur in two different configurations, L and D. Proteins are almost exclusively built from L amino acids. The stereochemical bias of nature is further reflected at the secondary structural level, where right-handed helices are strongly preferred over left-handed helices. The handedness of helix has not received much attention in the past and is often overlooked during the analysis, description, and deposition of experimentally solved protein structures. Therefore, an extensive survey of the left-handed helices in the protein data bank was undertaken to analyze their frequency of occurrence, length, amino acid composition, conservation, and possible structural or functional role. All left-handed helices of 4 or more residues in a non-redundant subset of the PDB were identified using hydrogen bonding analysis, comparison of related structures, and experimental electron density assessment to filter out likely suspicious and artifactual hits. This analysis yielded 31 verified left-handed helices in a set of 7,284 proteins. The 5 angles of the residues in the left-handed helices lie between 30 degree and 130 degree, and the psi angles lie between minus 50 degree and 100 degree. Most of the helices are short 4 residues, and for 87% of them it was possible to determine that they are important for the stability of the protein, for ligand binding, or as part of the active site. This suggests that even though left-handed helices are rare, when they do occur, they are structurally or functionally significant. 4 secondary structure assignment programs were tested for their ability to identify the handedness of the helix. Of these programs, only DSSP correctly assigned the handedness. This information you need not to know. The overall conclusion for this particular slide is that though both left and right alpha helices probability is there, but the more abundance is for the right-handed alpha helices, and we cannot completely remove the possibility of the occurrence of left-handed alpha helices. So whenever they are present, they do play an important functional as well as structural role. Okay. So what this is again coming to the very much basics of, we get too much deep into the secondary structures, but we should always remember why we want to understand the structures on the first hand, right? Why we want to understand about them, why there are 3D structures, why the primary structure of a protein is not functional, why it requires to have a 3D specific conformations. All these questions you should always keep asking these questions while studying the structure of proteins. Okay, so proteins are big molecules. The covalent backbone of a typical protein contains hundreds of individual bonds. Because free rotation is possible around many of these bonds, the protein can, in principle, assume a virtually uncountable number of spatial arrangements or conformations. In reality, however, each protein has a specific chemical or structural function which suggests that each has a unique 3-dimensional structure. So this is a very important statement. If we have a polypeptide chain, it has multiple bonds, right? So theoretically, there can be n number of arrangements of these bonds and angles, which can lead to different structures, right? But it is not the case. Since the beginning of the discussion of secondary structures of protein, I'm saying that there are alpha helices, beta sheets, right? So these are countable secondary structures. When there is a possibility of infinite number of secondary structures, we can clearly say that there are few structures which are more commonly present in the system. So there has to be some bias, or there has to be some restraint or some restriction which is limiting the availability of these infinite number of structures, right? So this happens because of the steric clashes. So though all the conformations of the atoms around the bond is possible, but due to energy clash or steric clashes, not all the structures are stable at room temperature or pressure, or say the conditions in which we are thriving. Okay, so only limited number of secondary structures are possible and they exist in the system. Protein structures are stabilized by non-covalent interactions and forces. Formation of a thermodynamically favorable structure depends on the influences of the hydrophobic effects, hydrogen bonds, ionic interactions, and van der Waals forces. Natural protein structures are constrained by peptide bonds whose configurations can be described by the dihedral angles. So, there are many non-covalent interactions which play a significant role in determining the structure of a protein. These are the forces which basically retain the 3D structure of a protein. It's not the covalent bond, though covalent bond are the strongest type of bonds, but in number non-covalent interactions are predominantly present, and these are the ones which actually stabilize the protein. So what sort of non-covalent interactions are present? They are hydrophobic effects, hydrogen bonds, ionic interactions, and van der Waals forces. If you remember our discussion of lipids, you know that these were the same non-covalent interactions which are responsible also for the stability of the membrane, right? So these forces, though weak in nature, but their abundance overcomes their strength. Okay, protein segments can adopt regular secondary structures such as the helix and the conformation. These structures are defined by the particular values of phi, and their formation is impacted by the amino acid composition of the segment. All of the phi and the psi values for a given protein structure can be visualized using Ramachandran plot. Okay, I guess I forgot to put the Ramachandran plot here, so just give me 2 minutes. Maybe I can quickly come up with the Ramachandran plot, and it will actually help us understand the.
In this video, we're going to
understand the possibility of these structures. I guess this is a good video. Let me share the screen so that you guys can understand the phi psi angles as well as Ramachandran plot. Yeah.
In this tutorial, we'll define the phi and psi angles of an amino acid. Use a 3D model to visualize them and see how they're measured. Finally, we'll learn how to translate what we see in the 3D model to a 2D Newman projection, which might sound familiar if you've taken an organic chemistry course. In order to carry out the functions that keep you alive, proteins fold up into very specific three-dimensional shapes. The peptide sequence curls and bends into the secondary structural elements of helices and sheets, and those can further fold into the tertiary structure of a protein. When crystallographers and structural biologists are figuring out the structures of these important macromolecules, they place the atoms where they appear to go in 3D space based on their structural data. But there can be some ambiguity in the model, so they need a way to verify that their structures make sense based on actual allowable molecular geometries. Enter the Ramachandran plot. In 1963, the Indian physicist G.N. Ramachandran described a standard way to define the geometry of a given amino acid in a polypeptide chain. Ramachandran's team noted that some geometries aren't possible due to sterics. In other words, a given amino acid cannot adopt some conformations in a polypeptide chain because of unfavorable crowding with the adjacent residues. And when the two backbone angles are plotted, there are distinct regions for the unique elements of the secondary structure. Phi and psi are examples of dihedral angles, which are, by definition, the angle between two intersecting planes, shown in yellow in this image. Notice that the carbonyl and the adjacent nitrogen are shown with dotted bonds. Remember that peptide bonds, and amide linkages in general, have significant contribution from a resonance structure with double bond character. So much so that the nitrogen and carbonyl group are coplanar. This puts 6 atoms in a single plane on each side of the alpha carbon. Though we don't have free rotation about amide bonds, we can rotate around each bond coming from the alpha carbon atom. Let's head over to our 3D model to explore this. We'll start with a tripeptide made up of 3 alanine residues. And for good measure, let's show the peptide bonds and label the N-terminus and C-terminus. When we measure phi and psi angles for our central alanine, we'll need to look at some atoms in the surrounding amino acids. But we can simplify our structure by removing atoms that are not needed to view phi or psi for the central alanine. Those atoms can obscure other atoms and make the analysis of the angle a little more difficult. The phi angle is defined by rotation about this bond, from the alpha carbon to nitrogen, which I've colored magenta. And the psi angle is given by the bond from the alpha carbon to the carbonyl, which I've colored green. In order to understand the angles, we need to set this up by looking straight at the alpha carbon directly down the bond of interest. So if we're looking for the phi angle, we'll orient the structure like this. And if we're considering the psi angle, we'll have to orient the structure like this. When we look down the bond for the phi angle, this nitrogen is going to be hidden behind the alpha carbon. What we'll be comparing is the angle between these 2 carbonyl carbon atoms. And when we look down the bond for our psi angle, this carbon atom is going to be hidden. So here we'll be comparing the angle between these 2 nitrogen atoms. So essentially, we consider a 4-atom pattern when looking at phi and psi. To translate this to our Newman projection, we can imagine the model as a clock. Looking directly down our bond of interest, we'll need to make sure to place the nitrogen or carbonyl carbon that's attached to the alpha carbon at noon. If we draw out just the front carbon of our Newman projection, this group has to point up. For the phi angle, our hydrogen points left and our methyl points right. When we draw out the front carbon atom for psi, we're looking at the alpha carbon from the opposite side. So our hydrogen points right and our methyl is on the left. Now it's time to consider what's going on with our back carbon. And we'll do this in detail for just the phi angle to start. Let's hide a couple more atoms to make the carbonyl group easier to see. Dihedral angles range in value from -180 to +180 degrees. If the 2 carbonyl carbons are eclipsed, the angle is 0. Let's start there and move incrementally to a phi angle of 180 degrees. Going back to our clock, as the carbonyl on the back carbon moves from noon to 6, the phi angles are positive. Let's pause at 90 degrees to draw the groups on the back carbon atom onto our Newman projection. The carbon of the carbonyl is
Hello everyone, myself Zeel Bhatt and welcome to my channel.
I guess this will be better. Because
Two
I wanted to
atoms
understand
cannot
the
occupy the
Ramachandran
same
part.
space. That simple principle determines a great deal of protein molecular structure. Most proteins consist almost entirely of alpha helices and beta sheets connected by turns and coil, shown here in white. It is the avoidance of clashes between atoms that forces proteins to adopt these common secondary and tertiary structures. To understand the Ramachandran principle, we'll use this small atomic model. In the center is a single complete alanine amino acid, all of whose atoms are marked by clouds of black dots. The atoms that are outside the black dots are fragments of adjacent amino acids that are connected to the central alanine in a polypeptide chain. Here is the main chain or backbone of the polypeptide, and over here is the side chain of the central alanine, the methyl group. The central alanine is connected to these other two amino acids by the two peptide bonds that are shown in magenta. In a polypeptide chain, each amino acid has two bonds that can rotate. Those bonds are part of the backbone or main chain. Each of those bonds defines a dihedral or torsion angle, something I'll explain next. Those two bonds are called phi and psi. A dihedral angle is determined by four atoms, numbered here 1 through 4. These particular four determine the phi angle. Notice the relative positions of atoms 1 and 4 when the angle is 0 degrees. Here we'll do a clockwise rotation around the phi bond. So we'll pass through 90 degrees where we're pausing for a moment. And then we'll continue arriving at a dihedral angle of 180 degrees. If we rotate counterclockwise around the phi bond, by convention the angles are considered to be negative. Here we're pausing at minus 90 degrees. And we'll continue rotating up to minus 135 degrees. If we continued to minus 180 degrees, that would be the same as plus 180 degrees. Peptide bonds have a double bonded character. This prevents them from rotating. They have this character because they resonate with the adjacent oxygen double bond. As a result of their inability to rotate, each peptide bond holds 6 atoms in a plane. In actuality, most phi or psi angles are impossible due to clashes between atoms. Remember that the atoms in this ball and stick model are much smaller than the true atom sizes. Here are the actual sizes of the atoms in our model. These are the so-called van der Waals radii. Here, the psi angle is fixed at 165 degrees, and we're rotating the phi angle a full turn through 360 degrees. As the atoms get close to each other and begin to overlap, which would be physically impossible, but is allowed in this model, They clash with each other, and these clashes are shown as the yellow and red objects that appear and disappear during rotation. Wherever these yellow and red objects appear, the clashes, that angle of phi would be impossible when psi is 165 degrees. Here is a Ramachandran plot. Along the bottom, we have the phi angles from 0 to plus 180 and 0 to minus 180. And along this axis, we have the psi angles from 0 to minus 180 and clockwise from 0 to plus 180. Inside the plot, we have 100,000 tiny dots. Each dot represents the phi and psi angles that actually occurred in a single amino acid in one of a large number of reliable protein structures. This cluster of dots represents alpha helices, while this cluster of dots represents beta strands that form beta sheets. And here we have turns. Notice that there are large areas of the plot completely devoid of dots. These conformations simply do not occur. Now let's compare our rotating phi animation with the Ramachandran plot. Remember that psi is fixed at 165 degrees. You'll notice that as phi rotates, there's only one region that has no clashes. Let's watch it. Here's a big clash, and just as it disappears, another clash, but starting here and going to there are no clashes. And that no clash region represents phi angles of minus 170 degrees to minus 70 degrees, namely the conformation of beta strands. Here is another animation in which we're rotating psi. In this animation, phi is fixed at minus 80 degrees. As you watch psi rotate, you see there are two regions with no clashes. The first one starts here and goes to here, and the second one is shorter, starting there and going to there. Those two areas of no clashes represent psi angles of minus 55 to 10 degrees, which is alpha helix, and 90 degrees to 180, slightly higher than 180.
Yeah, so you guys got an idea about what Ramachandran plot is. I was not planning for this for today, but as we are discussing the structure, so we should have an idea about the Ramachandran plot. Okay, getting back to our lecture, as I said that protein segments can adopt regular secondary structures such as helix and other conformations. This is because now we know that though there are infinite number of possibilities for the bonds to arrange themselves around the central atom, but only few configurations are permissible. Okay, hence we have very limited secondary structures. The tertiary structure describes the well-defined three-dimensional fold adopted by a protein. Protein structures are often built by combinatorial use of common protein folds or motifs. Quaternary structure describes the interaction between components of a multi-subunit assembly. We have already discussed this part. Okay. Tertiary structures is determined by amino acid sequence. Even though protein folding is complex, some denatured proteins can spontaneously refold into their active conformation based only on the chemical properties of their constituent amino acids. Cellular proteosis involves numerous pathways that regulate the folding, unfolding, and degradation of proteins. Many human diseases arise from protein misfolding and defects in proteosis. The three-dimensional structure of proteins can be defined. Structural biologists use a variety of instruments and computational methods to solve biomolecular structures. The choice of method may depend upon factors such as the size of the protein being studied, its properties, or the desired resolution of the final structure. The possible conformations of a protein or protein segment include any structural state it can achieve without breaking covalent bonds. So what are the conformations which are permissible? These are the ones which not gonna break the covalent bond, right? A change in conformation could occur, for example, by rotation about single bonds. However, of the many conformations that are theoretically possible in a protein containing hundreds of single bonds, only or a few generally predominate under biological conditions. The need for multiple stable conformations reflects the changes that must take place in most proteins as they bind to other molecules or catalyze reaction. The conformations existing under a given set of conditions are usually the ones that are thermodynamically the most stable, that is, having the lowest free energy. Proteins in any of their functional folded conformations are often called native proteins. Okay, so talking about the stability of a protein. Stability is the tendency of a protein to maintain a native conformation. Native proteins are only marginally stable. The delta G separating the folded and unfolded states in typical proteins under physiological conditions is in the range of only 5 to 65 kJ per mole. A given polypeptide chain can theoretically assume countless conformations, and as a result, the unfolded state of a protein is characterized by a high degree of conformational entropy. This entropy, along with the hydrogen bonding interactions of many groups in the polypeptide chain with the solvent, tends to maintain the unfolded state. The chemical interactions that counteract these effects and stabilize the native conformation include disulfide bonds and the weak interactions and forces. We are talking about secondary structure, tertiary structure, and overall structure of a protein, but with very less information about how to determine the structure of a protein. So we're gonna see what are the basic tools and techniques which are there in the field which actually predicts the structure of a protein. Okay.
This is a three-dimensional structure of a protein called lysozyme. It represents the order of nature, and I feel awed when I look at it. It's an enzyme that's in our tears and saliva and mucus, and it helps us fight bacteria. And knowing this three-dimensional structure helps us to understand the mechanism of action of this enzyme, and therefore how it helps fight bacteria in our bodies. Unfortunately, the size of the molecule is such that it's far too small for us to see under a light microscope or with our naked eye, because the wavelength of light is much, much larger than the size of this tiny molecule. For that reason, we have to use radiation